Good afternoon, everybody. It's always a special pleasure to welcome back one of our own. Gerardo was born, grew up, did his undergraduate in Mexico, but then was recruited by Carlos Castillo Chavez to come to Cornell for his PhD. Not in applied math. It was in biometry. It's one of those branches of applied math. It's big enough to have its own department, right? Graduating in 2005, and then going to a postdoc at Los Alamos lab with Mac Hyman, and from there to Arizona State University in this center with a very, very long name, where he has been ever since. If you haven't done it yet, I suggest that you go to Gerardo's webpage and just take a look at the breadth of what he's done. I think there are very few people out there who have published in the Journal of Mathematical Biology and in the New England Journal of Medicine, and who have a range of collaborators going from mathematicians out through people working in government health agencies on several different continents. So he's brought a very multifaceted approach to the study of infectious diseases, and I'm looking forward to hearing what he has to say today about seasonal and pandemic influenza. Gerardo.
Thank you, Steve, and thank you for the invitation. It's really very special to be here, to be back here. Thank you. All right. Okay, so this is my outline for my talk. So I'll start to by giving you a background on influenza. Okay. Then I will start talking about the 1918-1920 influenza pandemic, some of the work that we have done on trying to understand the epidemiological patterns and the timing of the spread of this pandemic. Then I will move on to the 2009 influenza pandemic, which we have been working with a number of people. And then I will talk about spatial transmission dynamics. I think that's going to be very interesting for a few students here. And then on seasonality patterns. And much of the work that I will talk about here is my work in collaboration with a number of people, and some work is actually also from others that I'm referring to. Okay. So influenza is a virus that spreads particularly in winter time in temperate areas. Okay. It affects about 5 to 15% of the population every year. Okay. And most of the severe cases are in the older population. So 90% of the deaths associated with the flu occur in people over 65. And one problem with influenza, working with influenza, is the clinical symptoms are not specific. So you have influenza-like illness, and that can be confused with other respiratory pathogens unless you do testing. Okay. So the other problem is that when we get a flu-like illness, we rarely go to look for help, and we are not counted in these statistics. And then if we go to the doctor, we may or may not be tested. So actually a very small fraction of influenza-like illness cases are actually tested for flu. And it's also the case that most of the severe cases are the result of secondary bacterial infections that could lead to hospitalization and death, mostly associated with individuals with chronic conditions like diabetes or some other diseases. And usually historically, we have relied on statistical methods to be able to quantify the burden and associated with the morbidity and mortality of influenza. Okay, so let's talk a little bit about the influenza virus. It's composed by 8 genes. Okay, so it's a relatively simple virus, and 2 surface antigens, so 2 surface proteins. The H, which is the hemagglutinin, that is the one that has the key to access the cell, and the N, the neuraminidase, that has the key to get out of the cell, so that other virions, new virions, are able to infect other cells in your body. Okay. So when a new subtype emerges, of influenza. Like one of these here, H1N1, H2N2, H3N2, which is the result of an adaptation of an avian influenza virus, okay, to the human population so that it becomes transmissible from person to person. Or maybe it's the result of a recombination of an avian influenza virus with an human influenza virus. But those are rare events, so we can expect that there are many new subtypes and strains that are generated in the avian population, in the swine population, but only a few of those actually are successful and jump into the human population. Okay. So the subtype H1N1, that particular strain that emerged in 1919, is the pandemic that we know as Spanish flu, generated about 20 to 100 million deaths in the world, about 675,000 deaths in the US, which translates into a death rate of 530 per 100,000. Okay? Then this subtype established in the population and continued to circulate in the population by making some genetic changes in the virus, relatively minor changes, that allow the virus to continue spreading in the population from 1918 all the way to the emergence of the next successful subtype, H2N2. This pandemic generated a lot less deaths, just about 60,000 deaths in the US, about 1 million in the world. Then you have H2N2 dominating transmission in the populations, basically all over the world. And you still have a little bit of transmission of H1N1. Then you have the H2N2 pandemic, so-called Hong Kong flu, that generated about 50,000 deaths. And then you have a reintroduction in 1977 of an old H1N1 strain, probably from a Russian lab, that circulated in the US and generated a lot of fear. There was actually a mass vaccination campaign that was started in this country, and that was able to vaccinate about 30% of the population. But that didn't really take off, and actually didn't generate many deaths. Then we have the recent 2009 pandemic that really generated important mortality, particularly in Latin American countries. This is what we are seeing so far, but not as much mortality as we had expected from the beginning of this pandemic. You see about 12,000 deaths, okay, in the US, compared to 36,000 deaths, which is the average number of deaths that we observe every year in the US due to seasonal flu, okay? Epidemic influenza over the last years that is associated with the H3N2 or the H1N1 or B influenza subtype. Okay. Okay. So this figure shows that pandemics actually spread in a number of waves. So in this graph, on the top left, you have the 1889 pandemic, where you see that London was affected by several waves. The number on the top of the curve is the actual percentage of the total mortality associated with influenza during that period of time. Okay. So you have here the 1918 pandemic, for example, with a summer wave here in Copenhagen, which was actually the pattern that was observed in many other places. Then you have a fall wave and a winter wave, and so forth. So just to show you that pandemics actually transmit in the population in a series of waves, and these waves can start at a time when you don't expect influenza to be transmitted, right? So they can actually start in spring and summer, where regular flu wouldn't be expected to be transmitted, but because a high fraction of the population is susceptible, you have a new virus, then the transmissibility is high enough and exceeds the threshold and is able to transmit in the population. Okay, so let's talk more about the 1918 influenza pandemic in New York. And this is work by Olson et al. In PNAS that showed that in 1918, you had the summer-spring wave, relatively small in New York, and most of the people affected were young people here, okay, and here. And those senior people, older people, than 65 actually enjoyed some protection to death, to mortality, as you can see here. The elevation here is very relatively small, particularly here, older than 65. And we have, or we think the consensus is that actually this 1918 influenza virus had been circulating back in the 19th century, probably in the mid-1800s, when these senior individuals in 1918 catch the flu or that related influenza strain during childhood, okay, and they will develop this immunity during childhood that is enjoyed later in life during this pandemic. So this pandemic virus 1918 is not quite like a new virus, but it's sort of recycled. And we will see that this is not the only pandemic in which we had this type of event. Actually, the 2009 influenza pandemic also showed that elderly sparing effect. This is data from Copenhagen. This is influenza illness and respiratory deaths that showed the summer, fall, and winter waves of spread. That I just showed you. Also, similar pattern for New York. The case fatality rate for the spring wave, for the summer wave, was 0.3% based on these data sets, which is lower than the case fatality rate during the fall winter. So the case fatality rate is the percentage of those people that catch the flu or report the flu that actually died. Okay? So it's not clear why this change in case fatality rate from the spring to the fall wave, whether it was a change in the virus, which is unlikely because it's just it's a short period of time. You don't expect this virus to evolve that quickly, or it was more of a host factor playing a role here. If we look at the age-specific mortality, okay, in Copenhagen, you see here 1910, 1917, that's the average curve, the classical U curve of mortality, right? Higher mortality rates among infants and higher mortality rates among senior and older people. And in 1918, you see clearly here that those young adults, 20 to 35 years, suffer disproportionately, okay, relative to the rest of the population. Okay, it's not clear exactly why this is the case, but this has been observed in a number of countries in the world. So it's really a pervasive pattern. Okay, and if we compute the relative risk of death, this is what you get, right? Higher mortality among young adults, and also the sparing effect among seniors. Okay. So we have, in these recent years, we have looked for this evidence in other populations, particularly in Latin America, and we have obtained data for central Mexico, where you can see here the spring, fall wave of the 1918 influenza pandemic. And by the way, these curves that you see here are just some curves that are used to set up a baseline mortality. It's the so-called statistical method self-link approach that has been used for a long time by CDC. And using those methods, we can estimate excess mortality by age groups. And what we see is that in Mexico, actually, senior populations and older populations experience high mortality rates, which is in contrast to reports from North America and Europe. Okay? And that's the case for Mexico City, and also for a close city to Mexico City, which is Toluca. But you still see here the high elevation in mortality rates among young populations. What about the spatial heterogeneity in mortality rates? We have looked at highly resolved spatial data from England and Wales. This is weekly mortality, influenza mortality for 305 administrative areas and for 62 counties. So each county contains a number of administrative areas to study how mortality rates could have actually differed between rural, urban areas and the pattern of spread in this country. So one of the findings that we found is that mortality rates actually were significantly different between rural and urban areas. So this plot here showed log-log transformations of the mortality rates across administrative areas, okay, among those urban areas compared to rural areas of England and Wales. And so if mortality rates, and this on the x-axis you have population size, okay, so these are organized by population size. If mortality rates were homogeneous across areas, you would expect a power law there, right, with an exponent of 1, but because mortality rates actually were lower among larger population rural areas, which are the red circles here. So you see this line here has a slope of about 0.7, and the blue line, which corresponds to urban areas, has a slope of about 1. So urban areas had quite consistent mortality rates across population sizes, but that was not the case among rural areas. So we think that actually death rates declined with population size in rural areas. So higher population rural areas probably had better access to care, right, that made them fare better during the pandemic period. But it's also interesting that if you actually work with the county level data, aggregate those administrative areas into counties, you actually don't see that effect. You would think that all England and Wales was equally affected, which is not the case. So that brings a lot of attention to the fact that working with spatially disaggregated data, it's always a good idea. And this shows that, perhaps not surprisingly, populations with the largest population size cities in England and Wales, like London, so forth, Sheffield, were the ones that experienced the pandemic first, the pandemic onset first, and then the pandemic virus was spread quickly after that through lower population areas, like in a hierarchical network of spread. So there's an insignificant correlation between population size and the timing of onset of the pandemic here in England and Wales. And this actually has implications for the calibration of transmission models for pandemic influenza. An important parameter in epidemiology in general is the basic reproduction number, which is defined classically as the average number of secondary cases that an infectious individual generates in a population on average, right, during his/her infectious period. And that's in the context of a completely susceptible population, but when we are talking about partially susceptible populations, that is, populations that have been affected before by a similar virus or perhaps by the same virus, and therefore you have a fraction that is resistant to infection, we talk about R, just reproduction number, just to make sure that that's clear. And so the estimation of the reproduction number has implications for determining the intensity and the type of interventions, public health interventions that will need to be put in place to control or mitigate the spread of an infectious disease. And R0 can be the simplest method to estimate reproduction numbers is just by looking at the growth rate of the time series of incidence cases, okay? And so this can be applied to flu or other infectious diseases, okay? So if we look at the rate of growth for pandemic influenza in England and Wales, 1918, these are the curves in log scale, okay? For the first wave, the spring, fall, and winter wave, and then you have the epidemics, flu epidemics that follow that pandemic, okay? And then you have this interesting 1950-51 epidemic associated with high transmissibility, but it's not labeled as a pandemic itself. And then you have the 1957 pandemic, the H2N2 that I mentioned earlier. So we see that for seasonal flu, the reproduction number is about 1.3 to 1.7. So there's some variability. And for pandemic influenza, if we take the 1918, we take the 1957 pandemic, the range is from 1.9 to 2.2. Okay? And so there are several reports on this. So we can look at the individual reproduction numbers by year on this graph here in England and Wales and in Liverpool. And we can see here the remarkably high transmissibility of the 1950-51 epidemic that my colleague Cecile Bivot noted and published in Vaccine a few years ago. And we have also looked at more recent data from the US, France, and Australia. Yes, question?
So with other transmissible diseases like STDs, in the popular science articles I read about these super spreaders, that the reproduction number is high not because everybody produces a large number of secondary infections, but there's a small fraction of the population produce a huge number of secondary infections. With influenza, is it known whether the pandemics have high R because of a small population of super spreaders, or is it an inflated transmissibility throughout the population?
Yeah, they play a role, but it's not clear whether they drive all these epidemics. I think the evidence or the data that we have on contact networks and that has been collected during the pandemic period doesn't quite show that super spreaders are the ones that contribute to 80% of the transmission, the 20/80 rule, for example, right? But I think this applies more to, as you said, sexually transmitted diseases. Yeah. But I have to say, there's limited data, right? We need more data, and perhaps in some situations, the role of super spreaders is higher than in others. As you will see, there are many other factors that are playing a role in transmission of influenza. Okay. And so what we saw here is that transmissibility is quite consistent across these three countries, the US, France, and Australia. Remarkably, the variability is important also in all of them, but the reproduction number is small, and it's just 1.3, that you would expect that some intervention campaigns should be able to control the spread of influenza, right? But it's not working. Our vaccination strategies are not quite working. There are some problems, right? Okay, so I was like lucky, perhaps, that I went to Mexico City to work with the Mexican government via Fogarty International Center at the very early phase of the 2009 influenza pandemic to try to understand what was going on, to try to understand the growth rate of the pandemic, what were the highest risk groups, and why there were a significant number of young adults that were hospitalized, and some of them actually were dying. So it was an interesting time. And one of the first findings that we reported is that actually young adults, again, you would expect from past pandemics, were being hard hit, as you can see here in red. The proportion of deaths was focused on those young people, and this is just during the early phase of the pandemic, which is in contrast with the sort of U-shaped mortality curve that you would obtain from the data from Mexico for the last two seasons, so the last 2 years. Okay, so this would imply that perhaps control interventions should be focused on young people, in vaccines in limited quantities, if available, should be then deployed on this age group. Then, as more data became available, this is data from the Mexican Institute of Social Security in Mexico, probably the best data available for H1N1, because this is a surveillance system that covers about 40% of the population of Mexico. Okay, so these are people that are insured with this government subsidized system, and so all the persons that go to the doctor to the IMSS, healthcare facilities, hospitals, and so forth, are reported in this system. Okay? So you see there clearly the spring wave that was actually limited by a school closure. Okay? So when the Mexican government noted that there was a significant number of young adults that were being hospitalized and were dying, they decided to close schools first in Mexico City and 3 days later all over the country. And that seems to have had an impact, and I will show you some analysis that we did in the next slide. Then we have pandemic flu resurging in the southeastern states, okay, of Mexico. And then by the time summer vacations started, the pandemic started to decline again. And this makes all sense because we know that school-age children play an important role in the dissemination of the influenza virus. And then you have this big wave in the fall when kids start to go back to school again, right? It was a more generalized way. Okay, so if we look at the role of the school closure in Mexico, this shaded area here corresponds to the spring break, okay, that time. So we can measure the reproduction number just looking at the incidence during that time from the data that I just showed you. That would give you an R of 1.3. Then you have the end of the spring break. And by the way, this time scale is relative to the onset of symptoms. Okay, so we are noting here incidences at the time, number of new cases at the time of symptoms onset, which is important. Okay. Then you have a reproduction number of 2.2 here, and then by the time that the school closure started to take place on April 24th, you have this saturating effect, which would suggest or indicate that the school intervention was having an impact. Okay, and actually by the end of that school closure, the trend continued to go down, which is also interesting. So at this point, we know that school closures, we put all the evidence together, they have an impact on reducing incidence of influenza, at least temporarily, but it's not clear how long these school closures should last and when they should start if we want to control this flu spread. Okay? What about the mortality impact of the pandemic? We have looked at excess mortality in Mexico, as you can see here. This corresponds to Mexico, all ages, people younger than 65, and this is people older than 65. You can see here excess deaths, and this is by doing a statistical model, right? The statistical model defines this baseline of mortality in gray, so that everything that is above that baseline is considered to be associated with influenza. Okay. So what we found is that in Mexico there were 13 deaths per 100,000 population. That was the mortality rate. While in developed countries like the US or France, you had only about 1 death per 100,000 population. And actually looking at data for other Latin American countries, overall it looks like Latin America had the highest mortality rates, okay? Although there's no data for Africa at this point to measure excess mortality. We can also look more closely at the pattern of the case fatality ratio, that is what fraction of those that got infected, that reported influenza, that actually died over time during the pandemic in Mexico. So the top graph shows the H1N1 positive outpatients and H1N1 positive inpatients. Okay. This graph here in the middle shows antiviral treatment. So really, antiviral treatment was used quite a lot during the early phases of the pandemic and reached about 75-80% antiviral treatment. So that would mean that every person that would come to the health facility, they would get antiviral with that probability, okay, 85% maximum, 75%, okay, and there was no difference whether it was a patient that went to an outpatient facility or actually was admitted into the hospital, which is interesting, okay. But it's most interesting that after the hype, the first few months of the pandemic, antiviral treatment seems to have gone to very low levels, like 9%. And at the same time, the case fatality rate started to increase, as you can see here in the bottom graph. Okay? So it suggests that actually this behavioral pattern in the medical treatment had an impact on the case fatality rate in Mexico, and probably could explain part of the higher burden of mortality that we are seeing. We also noted that admission delays less than 2 days. So once your symptoms start, if you go to the hospital within 2 days, that would decrease your mortality risk by 50 to 75%. So if you start to feel bad, it's better to go earlier than later. Antiviral treatment reduced mortality by 48%. Okay, and this was done adjusting by age, by gender, pandemic wave, and geographic region in Mexico. Okay, that was the best data available. There was no adjustment for other underlying medical conditions for this analysis. Okay, so now let's move to Chile, where we had access to an interesting data set. And Chile is a very unique country, as you know. It spans a very large latitudinal gradient from north to south. And what we noted here, trying to understand the dynamics of influenza by looking at different data sets from all over the place, we noted that there is a gradient here from south to north, such that transmission of influenza occurred faster in the lower, in the southern regions of Chile, and then move up to the center and the north Chile. Okay, even though most of the population is located in the capital in Santiago. Okay, so you see here that there were introductions in Santiago of flu H1N1, but it didn't really take off at that time. Okay, it really took off later on, as you can see here, and the transmission occurred first in the southern region. So there's a gradient, and the gradient is significant. So we're trying to understand why this gradient is actually going on, because population size doesn't explain it, or international travelers arriving across the airports in Chile doesn't explain it either. So we decided to start looking at actually climatological variables, given the large latitudinal gradient there. So we saw, interestingly, that we expect an important gradient in temperature from north to south Chile. No really interesting pattern in precipitation. Okay, so what you are seeing here is precipitation in different months during the pandemic from north to south Chile. Okay. For relative humidity, it's quite noisy also, no clear pattern. And for a specific humidity, there's also interesting pattern, interesting gradient there, just like for temperature. Okay, so we take these relationships and correlate them with the peak timing across regions in Chile, and we found that yes, there's a correlation there, even after you adjust for population size or for international travel passengers or number of seaport landfalls across regions. Okay, and this goes very well with the fact that we know that flu is more efficiently transmitted at lower temperatures and at lower specific humidity levels as well, or in general humidity. The experimental studies have been done at different ranges of temperature and humidity levels. Okay, and this finding is also corroborated by the fact that the growth rate was also different across regions, higher in the south, so you have higher transmissibility. That means the epidemic is expected to peak earlier, followed by the central part, and then after the north region. So there's a gradient there also in transmissibility. Okay. So what we can summarize so far is that the 2009 pandemic was characterized by mortality age shift, okay, with young adults, just like in historical pandemics, young adults being hardest hit, particularly for severe disease and mortality. A moderate reproduction number of 1.4 to 1.5, which is closely in line with that of seasonal influenza. It was low to moderate severity. Of course, the true case fatality rate is difficult to measure because we never have the exact counts of how many people were infected with flu during the pandemic. We saw important geographical variation in mortality burden, particularly in Latin America, higher mortality rates, probably associated in part with the use of antivirals, particularly in Mexico. We saw that school closure can decrease transmission of pandemic influenza, particularly if it is implemented early. But the exact timing and duration of these school closures has not been well established from models that actually get connected to real data, right? Because we can model these things and find some results, but if we can actually validate them with the data, that has not really occurred yet. As a summary, the signature features of influenza pandemics, we found that there are multiple waves of various severities, higher transmissibility for pandemics than seasonal influenza, shift of influenza-related deaths toward people that are young adults particularly, and the sparing of seniors has been observed in North America and some areas of Europe, but that's not the case for Mexico, that's not the case for other settings in Latin America like Colombia and Chile that we have also explored. And that could be explained by the fact that in the 19th century, travel was much less developed at the time, and so the circulation patterns of influenza were not quite homogeneous, but more heterogeneous. So perhaps populations in North America and Europe that were more connected were more exposed to influenza viruses that gave them some protection to the pandemic in 1918. And we also noted this spatial heterogeneity in mortality rates during pandemics. Okay, so let me talk a little bit about spatial transmission dynamics. And this is work by Cecile Vivaud, 2006, where they used data, 30 years of data for the US across states, the US, to validate a metapopulation model based on a gravity force of infection, gravity model, they call it gravity model for workflows, okay, that is calibrated against this nice data set across states of the US, okay? So they found that these workflows calibrated through this metapopulation equation work very well. So you take into account the population of one state, counties actually, these are counties. And then you take the population of another state, and then you divide that by the distance, like a gravity equation. And then you look for the best exponents here that gives you the best epidemic phase correlation relative to the workflows between all the areas of the US. And they found that this model was a good tool to model the spread of influenza in the United States, such that when you take this model, you can start playing with transmission patterns. So you can start asking questions such as, what happens if influenza starts in California? Where are the places that are most likely to be hit after that? And this is the case for California, this is the case for Wyoming, okay? And so you can generate these transmission patterns across the US. More recently, my colleagues have been looking at data for the 2009 influenza pandemic at the zip code level, okay, to look at the spread by county, actually. And so these are all the coverage of this surveillance data. This is an influenza-like illness surveillance system. And if we look at New York State, that just highlights the fact that there is important heterogeneity in the transmission. the transmission patterns. So some areas of New York, actually like New York City, you had a large spring wave that was actually larger than the fall wave. It's interesting. But there were other areas, probably less connected areas to New York City, that didn't quite experience a spring wave, but had a large fall wave. Okay? So how can we take all this data into a model that could help us understand the spread of flu in a big country like the United States? If we look at the timing of onset of the pandemic across the US, we can see that there were definitely important temporal variations. So you have Atlanta reaching a peak in the pandemic first, and then you have places like Boston or New York with a late peak. Okay, and we're talking here about the main pandemic wave in the fall. Okay, so if we take a model like the model developed by Cecile Bivot, but now we can include more factors, because we know that temperature or specific humidity play an important role. We know that school closures play an important role. So how can we expand this model to better describe the pattern of spread? So here we have the force of infection here in this big equation, and I'll just show you what it means, each term here. So we have a term that modulates transmission with the humidity. Okay, so humidity will be varying over the pandemic period, and we know that it can have an important impact on transmission. We have a school open indicators to know whether the school was in session or not, because if the school is in session, then we can expect transmission from school-age children, right? And then we have population size from the donor and recipient, right? So this is the transmission route or direction from county A to county B, and then we have the distance between these two counties, and a number of exponents that you can see here that are scaling factors, okay? That are the ones that are going to be tuned, okay, to be able to find the best fit of this model to the data, okay? So it turns out that the best model actually has a small seeding, so a small number of infections that are needed as a baseline for transmission. There's weak dependence on the recipient population size. There's a strong spatial dependence, okay? That plays an important role. The best fit model. And then the schools are important, but their effect actually is not that large, okay? Which is a little bit surprising, I would say. Okay, so these are the results. This is just the model fit when you include only the school cycle timing. So when the schools are in session or not, and you see that the model doesn't fit data at all. And this is the model that I just described, okay? This is the model with all the factors that I just showed you. And this is what gives the best fit here. But there's still work to be done to better describe this data set. Okay, if we want to sort of validate these findings here, we can use molecular epidemiology, pandemic influenza data. And it turns out that there were 3 cities in the US that actually boosted their testing rates, which is great. There were Houston, Milwaukee, and New York State. And these are the patterns just from those sites for all the visits that were coded as influenza. And a high fraction of those actually were tested for flu. And these are the molecular epidemiology result, the phylogenetic trees. This is for the spring wave, and this is for the fall wave. And what Nelson et al. Found is that the spring wave, you had actually several lineages of the H1N1 influenza virus circulating. Okay. By the time of the fall wave here, there was basically one single lineage that was being transmitted all over the place. And this model, actually molecular epidemiology model, indicates that there was no important spatial structure here, which is in contrast to the finding in the previous slide, where it finds that distance is very important. So those are things that are still being revised. So far, according to these model efforts, this indicates that there is a very local spread, very important local spread. The timing of the fall wave doesn't appear to depend on humidity, which is also surprising. On the presence or absence of the spring wave, which confers immunity to people prior to the fall wave, or the size of the donor population size. And as I said, the school term played a minor role, and there is some inconsistency then with the molecular epidemiology data. So not everything is beautiful, right? And then also this model predicted spread was slower than it was expected actually for this pandemic influenza. Okay, what about seasonality? Seasonality is an important area of research for influenza, trying to understand why different places around the world actually observe different peak timing of flu at different times of the year. So for example, you have here Bismarck in the US, where the peak timing occurs during the winter. Most of the incidents are average in December. Then you have Singapore with two peaks, right? December, January, but you also have here another peak in the middle, Singapore in the tropical belt. You have places like Fortaleza, Brazil, with very good data that shows here some sort of spring peak timing of flu. And then you have Sydney, Australia in the southern hemisphere here with a winter time epidemic of influenza, right? So what model that connects climatological variables could explain these differences in peak time? Right? So we can look at the probability of an influenza peak, okay, occurring by month, which is the x-axis, and latitude, which is the y-axis, based on all the studies that have reported peak timing of influenza in different parts of the world. And that includes about 78 locations. And as you can see, there is less data for these tropical areas here. But you can see clearly here, this cluster here, okay, the winter time in the northern hemisphere, and then you have here this cluster here in August, but it's less strong, the signal there, and then the southern hemisphere here in the winter time epidemics. So how can we take all of this and get a model that could explain these patterns? And so what Tamarius has done is to look actually at a specific humidity, okay? A specific humidity in relation to the peak timing of flu across all these 78 locations. And what he has found is that actually the relationship is sort of bimodal here. So the probability of having an influenza peak is high when you are at low specific humidity regimes, but also is high when you are at very high low specific humidity regimes. And if you use that model, you can sort of predict the timing of influenza season across all over the world. And the model does better, obviously, as you move to the northern region, the northern hemisphere and the southern hemisphere. And for the tropical belt, where you have less variability, right? Obviously, less variability in specific humidity, it's harder to predict when this influenza peak will occur. Okay, so as far as influenza seasonality, we can summarize that low humidity, as I had mentioned, favors transmission in laboratory settings. There's a bimodal relationship between transmission and humidity that is evident from this epidemiological data. And as I said, it's difficult to predict the tropical belt, but perhaps for the tropical belt, actual contact patterns between people play a major role than the actual temperature, specific humidity levels. And it would be interesting, I think, as a future work, not only to look at influenza, but to look at other respiratory pathogens with similar transmission mechanisms like respiratory syncytial virus or other adenoviruses. And finally, I would like to acknowledge that this work is in collaboration with a number of colleagues, particularly my colleagues at NIH, Cecile Bivol, Mark Miller, and Vladimir Alonso, and Lona Simonsen. Thank you very much.
We have time for some questions.
So at the very end, you were showing that it's harder to predict the peak location close to the equator. But so it was a little bit unclear to me from your earlier graphs. So are the peak magnitudes close to the equator of similar amplitude to those in the farther latitudes?
Question. It's actually lower magnitude. The seasonality of flu in the tropical areas looks more like flat. So it appears that flu is circulating, at least from studies from Brazil, from my colleague Vladimir Alonso. It appears that influenza is being transmitted all over, all the year round in tropical areas like Brazil. If you look at areas in the North Brazil, you see that flu is being detected all the time, which is interesting. Yes?
You seem to suggest that it might be possible to prevent flu epidemics in the U.S. and some of the European countries. Do you have suggestions about how to do that?
Okay, yes, I have a few. I just talk to you. I think we have a vaccine in place, right? Yeah. And the vaccine doesn't seem to be working well. That's right. So, for example, the last report for the seasonal influenza vaccine for this year shows that it has no effect at all on senior individuals. No effect. So, sorry if you took the vaccine, it didn't work. Actually, the confidence interval goes to negative numbers, which is embarrassing. For young individuals, it's working moderately well, with an efficacy of about 50%. And that's conditional on having a good matched vaccine, which actually happened this year. Even under those conditions, we still have a vaccine with limited effectiveness. So the vaccine, we need a better vaccine. And I think also we need to vaccinate people earlier and earlier. For example, in this season, we had a very early onset, okay, particularly in the Northeast, at a time when we were still vaccinating people, right? I didn't present that work, but my colleague Sherry Towers and I and others have shown that actually the transmissibility of this season, right, the actual season, is associated with the temperature the season before. So if you had a warmer season, the previous epidemic, you can expect, on average, higher transmissibility this season here. Which is, I think, is some progress, because up to this point, we have not been able to predict neither the attack rate or the onset of flu epidemics for the next season. So there's a lot of work to be done in that regard. But I think it should be possible to control influenza. We can do much better. Yes?
Why do you think the peaks are less strong in the equatorial region?
I think temperature and humidity are two very important ones. High temperatures are reducing the transmissibility of the flu. So from having an reproduction number of 1.3, that probably goes down to 1.1, very close to threshold. So I think those are the major.
But as new strains are introduced every year, you would expect there to be peaks nevertheless, right? But we don't see those peaks, right? So
Right. Well,
it's because of that. So in other
no,
words,
actually, there's no new strain necessarily being introduced every year. Usually, you have a strain that circulates and sweeps the population for 3-4 years, and then you have some genetic mutation, minor, that generates a new strain. The real new viruses occur during pandemics, the new subtypes. But usually during 5-year windows, you have basically the same strain just going through the population many times.
so you think it's more the temperature and humidity rather than the fact that people are sort of always exposed to it, so they sort of keep up their immunity. So how you
Yeah.
separate
I see what you're saying. Yeah, it's difficult to disentangle. So you're saying because influenza is actually being transmitted continuously, then there's higher susceptibility, higher protective levels in the populations. That could work well actually, and we have argued on that one too in some papers. So that's good. That's a good one. And that also implies that if you have higher protective levels, you may be pushing the virus to change more rapidly, right? That should go in line with that, and therefore you would expect new strains emerging from tropical areas, which is the sourcing model that has been published, but recent studies are not quite supporting that, right? And the pandemic in Mexico didn't support that either. So lots of things to think about. Thank you. Yes.
I'm curious, because you're on the interface with public health, supposing we had a really good model that explained the spatial and temporal variability in attack rates, do you have a sense of what your colleagues would do with that? Would it be targeted distribution of vaccine? Or how in practice would a really good predictive capacity for influenza lead to its eradication?
Right. So definitely, I think that would inform your control strategies. That's the first basic step, right? Where you should deploy vaccines first if you have a limited quantity, for example, because not all the years we have enough vaccines. Sometimes we have shortages. So if we can understand where flu starts to spread first, and which populations actually experience the earliest onset, then it's almost right direct implication there. And then if we can understand better how environmental controlling the temperature in rooms, for example, right, to control flu. I know some people control their humidity levels in their households, and they think it works. But I think we need more studies, and there are work actually being done by people like Jeff Chaman at the School of Public Health in Columbia University, who is actually doing field work, measuring these numbers in different rooms in different places, so that we can understand better the spread of influenza. So.
For those.
Oh, sorry.
Yeah.
That's it.
Alex has been waiting.
Okay, I'm
Oh,
sorry.
okay.
Yeah.
I have a quick question about the gravity model which you have presented there.
You want me to move?
So, that's okay.
Okay.
I mean,
Okay.
you're fixing the
General.
exponents based on data. Is there some simple explanation on an agent-based behavior? Right? If I were to build an agent-based model, what kind of behavior of that agents would result in those exponents? Is there an interpretation?
Oh.
What
I
kind
don't
of
think it's
stochastic
separate.
interactions?
It's difficult to interpret these exponents. Every time you put these power laws, it's difficult, right? But if you use if you have an agent-based model, that means that you have a handle on all the individuals in the population, and then you can have complete control over.
Yeah, but I need to build somehow how they're
You
going
cannot
to
go from
interact.
this model
So-
to an agent-based model.
So I guess what I'm asking is there some elementary way to build their interactions so that those exponents would fall out of that?
Yeah, I wouldn't take that approach. right? If you're going to use an agent-based model, then you have to build from first principles, not take this higher level model back into the agent-based model, in my view, right? But we can talk more about that.
If there's one more question, let's take it.
Just for those of us who are not as practiced at thinking about these mechanisms, and just out of curiosity, how would higher levels of protection in the population put an evolutionary pressure on the virus to evolve faster? Say in the equatorial areas that we heard about.
That's what you would expect from evolutionary theory, right, Steve? What do you think? How would that be?
Yeah, well, it would mean that an escape mutant would have a much higher ratio of susceptible, right, under those circumstances. But it sort of means that there's a large population out there that's only accessible, okay, to a mutant. So, whereas there'd be a larger advantage if you can get around the immunity that's out there.
And
That makes sense off the top of my head. Whether it makes sense in half an hour or not, I'm not sure.
right. I mean, that's a very short response, and there are also very detailed evolutionary models of influenza, where you actually model the spread of a number of strains, and you see what happens in the long term, right? Taking into account the susceptibility, cross immunity of different strains, differential transmissibility, and so forth.
All right, let's thank Gerardo again and move on to the refreshments down the hall where we can continue.